Okay, so the point we left off, we had generated the 2-pyruvic acid, and if needed, 2 ATP that the body can use. Now, if the body is not being in a position where we're like throwing out our oxygen faster than we can utilize it, okay, and we're just simply like we're doing here right now, we're going to move into the aerobic respiration. But at the point where the glycolysis ended and we produced only the 2 ATP, that is our anaerobic respiration or fermentation, meaning there was no oxygen present. We did produce energy, very little, and we ended up with a huge molecule of lactic acid. Now, if we move into the aerobic respiration means we're now going to need an oxygen molecule. What do? We breathe, okay? And the oxygen that we breathe has a purpose. It's not just like superfluous, it's there for a reason. So aerobic respiration, this now means we're going to take that glucose that we split into the 2-pyruvic acid that's going to enter into the mitochondria with the help of an oxygen, break it down, strip a bunch of electrons off of it. And that means if we strip a bunch of electrons off of it, we got to deal with those little hydrogens, okay? And we're going to shuttle those electrons to the electron transport chain. That is the goal. Get the electrons to the electron transport chain. So we've now got those 2 pyruvic acids. We're going to enter into the mitochondria. We're going to go through the citric acid cycle, and I've been calling it Calvin. I'm sorry. Calvin is in the plants. I'm sorry. Citric acid cycle. I just like Krebs, okay? But somebody decided years ago to go to this, and I just liked Krebs. And our electron transport chain. Now, I do not in any way expect you to know the steps that we're going to look at right now, okay? I'm simply showing you there are lots of steps that exist. The reason those steps exist, every time that pyruvic molecule went in and it has to go through one of these steps, it changes its shape and we get to strip off electrons. Because the goal, get the electrons. Does that make sense? All right, so we end up with, and I want to do it without messing up the camera tries to get me and I move too much. I don't stand still, so I guess I'm going to have to go up there and be boring and stand still. All right, so we ended up with our 2 pyruvic acid molecules. They enter into the electron, into the mitochondria. When they enter into the mitochondria, we strip off a carbon. So now it ends up being 2 carbon. That 1 carbon that got stripped off, we're going to see that we get our production of our CO2. The stuff that we breathe out. Okay? Now, the rest of that carbon chain enters into the Krebs cycle or the citric acid cycle. And it enters, things get attached, things get broken off. Okay? Like I said, I don't care that you know these steps. All right? What I'm trying to get you to focus on is that every time that cycle begins, there's a step that's going to occur, and we're going to get something. Like, for example, we're getting the CO2. Another step occurs. What do you see right here? We see NAD, FAD. What did we say they were? Electron carriers. So at this point, electrons got stripped off because it became NADH, FADH. Look at how many I got. That looks like a lot, right? That means all 6, for example, all 6 of those cars shuffled to the electron transport chain and drop off those electrons in the H pluses. Everybody with me? So each time, this is what's happening. No. Okay? But now let's look at what our results are. Okay? So I entered, something gets taken, something gets added, stuff gets taken away. As I move through this chain, look, at each little step where we see something occurring, I got some CO2 coming off, I've got some NADs, I've got some FADs, I've got some ATPs, I'm back up here, and this process will start all over again with that second pyruvic acid that got created. So that second pyruvic acid will come through, go through all of these little steps, we'll strip off those electrons, okay, and now we get to shuttle all of those electrons to the electron transport chain. It's at the electron transport chain we get to generate roughly the 32 ATPs. Now glycolysis only gave us 2. I'm now generating roughly 32 ATPs. Which one seems a little more efficient? Aerobic. Way more efficient. Gives us producing roughly those 32 ATPs would be the equivalent of somebody giving me $1 million. Think about that for the cell. Okay? That's how efficient this is, how important this process is. Now, something I got to point out, okay? The structure of the mitochondria. Remember, we do nothing to control the mitochondria, okay? Mitochondria take care of themselves. They have their own DNA. They duplicate when they're told to duplicate. We do nothing. They have a bi-layer, so that tells me they are 2 layers. Based upon the structure, the outer layer is that one huge loopy thing around all of the squiggly lines on the inside. So we have that outer layer and the inner layer for those 2, the double membrane that exists. Which one do you find the electron transport chain embedded in? The inside or the inner membrane. Now, if I have a situation where it's a closed space, like the mitochondria, where I've got a membrane that goes all the way around, and then I've got this little inner folding membrane system, does that create like a closed-in area around or between the 2 membranes? In other words, is it like a separation between the inner to the outer? A space? Yes. I've got a space that's present between the inner membrane and the outer membrane. Now, everything that's being shuttled to the electron transport chain all of those little proteins and everything are in the inner membrane. The electrons get shuttled to the first protein, and then that electron begins to bounce down all of those proteins. What do you see the H pluses doing? Because remember, we balance the electrons, which are negative, with the H plus. What do you see the H pluses doing? They are getting trapped between the inner and the outer membrane. The final, well, right here, where we're seeing H pluses, H pluses, H pluses, because every single time we bump down a protein, H pluses that got drugged there get to go across the membrane, but they can't come back across. The only way they can come back across is through this protein right here called ATP synthase. What is it producing? ATP. So what we end up getting between the space here a concentration gradient of the H pluses. And materials like to move in which direction? From an area of high to low. So if I build up a concentration gradient of the H pluses as those electrons bump down that electron transport chain, the only way for the H plus to cross back over is to flow down the synthase, and when it flows down, it gets converted to ATP. The final electron acceptor, as we bump those electrons down this chain, when it gets to the last protein, the final electron acceptor is oxygen. That's where we need the oxygen for aerobic respiration. That final electron in the chain. Oxygen attaches to it. As the hydrogens flow down the ATP synthase, we have now generated the ATP. For every glucose molecule, because we created 2 pyruvic acids that went through the process, for each one that's going to bump down the electrons with the H's going across and the H's coming back down, we generate roughly the 32 to 34 ATPs. That's how the cell gets energy, which is a really cool process. So if we look at this overall production from one glucose molecule, if it goes through all the steps, in glycolysis, we generated roughly 2 ATPs. As we go through the breaking it down from 3 to 2 carbons, we got roughly 6 ATPs. As we put it through the citric acid cycle, that generates another 24 for a total of roughly 36 ATPs for every glucose molecule that we use in the body. Now, what does that mean energy-wise? Energy-wise, when you do, say for example, a strenuous exercise, like maybe mountain biking. Y'all know anybody who mountain bikes? Okay, that's a very, I wouldn't do it. Okay, it's a very tough sport to do. okay? For every second, a single cell of their muscles in their legs need about 10,000 ATPs. That lets you know how important the production is of ATPs. This is the process that we get it. No questions.